Turgor pressure. When a cell is placed in water or a hypotonic solution, the cell absorbs water because of the higher concentration of the cell sap. When water enters into the cell, naturally inside the pressure builds up, and this pressure is exerted on the wall. And if the cell wall is elastic, naturally the cell increases in size. It is because of the pressure exerted by the cell solution. That pressure we call it Turgor pressure. Turgor pressure is denoted by a symbol psi p. Now, this cell is said to be a turgid cell. In a plant, the young stems stand erect, the leaves unfold, they open and they are stiff. It is all due to the Turgor pressure. A turgid cell has the Turgor pressure. If there is no Turgor pressure, that means the cell inside the protoplasm in an isotonic solution, means the concentration inside and concentration outside are equal. The cell will not absorb water and the cell will not become turgid. The cell retains its original size. The cell wall remains in its normal position. There is neither Turgor pressure nor wall pressure. Then this cell is said to be in flaccid condition. So, this cell is described as flaccid cell. If the concentration outside is more, hypertonic, then the water from the cell comes out, plasmolysis, and that cell where the protoplasm has shrunk, we call it plasmolysed cell. So, the cells are in three states: turgid state, where the cell expands; flaccid state, where there is neither external pressure nor internal pressure; plasmolysed, means there is loss of water from the cell. So, these three states can be observed in plants. When the seeds are dried, naturally the percentage of water inside the dry seeds is only 5 to 10%. That means the cells are plasmolized. In living condition, in a living plant, you do not observe the plasmolized condition, but in dry states you can observe it because we dry them.